Hello, I'm Ting from Childmind Institute. It's a great pleasure to have this opportunity to share the work at this gradient workshop. I'm going to present cross-species functional alignments based on the gradients and to show how it can review the evolutionary hierarchy within the connectome. The cross-species comparison is very important for understanding the human brain organization and the brain evolution. For example, previous studies have shown that evolutionary cortical expansion from macaque to human has very similar pattern compared to the childhood cortical expansion. It also has very similar pattern with the inter-individual variation and long-distance connectivity distribution in humans. In response, it is essential to establish the alignment between species for such comparison. And this can also facilitate the translational efforts in animal models. The benchmark alignments in last decades was proposed by David Van Essen. This approach made use of common recognized anatomical landmarks to register cortical surface from one species to another. The more recent efforts from Rogerma's group made use of white matter tracks and myelination to align non-human primates to humans. One recent effort started to use functional data while human and macaque are both watching the same movie to align them together by comparing the neural activity during watching movie. But what else of brain function is comparable for us to do the alignments? So the answer might be gradients. If we can identify comparable gradients between macaque and human, then we can align them together. And this seems to be possible based on Daniel Markley's gradient work, showing that human and macaque brain have similar hierarchy gradients along the surface. So here, I use two public available datasets to do the analysis. For human, it's Human Connectome Projects, and for macaque monkey, it's Primate Data Exchange Consortium. Instead of using diffusion embedding for each species to match the gradient afterwards, here we propose a joint-embedding approach, which can extract the matched gradients in human and macaque simultaneously. So how do we do that? We start with this gradient, anatomy landmarks that may be established in David Van Essen and Rogerma's work. And then we first calculate the vertex to landmark functional connectivity within species, and use this vertex to landmark connectivity profile to get the human connectome of human macaque similarity matrix. Then we can build a joint similarity matrix to place the within species similarity on the diagonal part and the between species similarity on the off-diagonal part. We can perform the diffusion embedding algorithm on this joint similarity matrix to extract the components. We can also call the components gradient here. And the first half of the gradients were matched with the human vertices, and the second half of the gradient matched with the monkey vertices. Then we can use each gradient or these components as one dimension to build a high dimensional space. So here is the first dimension. Each node in human and macaque can be projected in this high dimensional space. And each node also corresponds to each of the cortical vertices on the human and macaque surface. As you can see here, they align quite well in high dimensional space and being on the surface. This is the second dimension. highlights the gradient from sensory motor regions to high order associated cortex. The third dimension. Now we show how it can be aligned in this space. In this way, we can link human brain and macaque brain together. Here's an example, we mapped the glasser pixelation from human to macaque brain. And finally, we can simulate the evolution of surface expansion from macaque to human. One thing I didn't really include in the animation is the surface matching process. Let's record this gradients for a second. Once we put the gradient on the surface, we can actually align the gradients, use this gradient as a cortical feature to align them in MSM, the multimodal surface matching. So here we show that alignment can align human mind map, which is T1 divided by T2, and map into the monkey surface and compare to the real T1 divided by T2 calculated in the monkey space. So we see that these two mind map is quite comparable. So to demonstrate the alignment works. Based on this functional cross-species alignments, we can also map the human population into the macaque surface, and another way around from the macaque population map back to the human surface. We can also map functional connectivity between human and macaque. Here is an example that we place the seeds in human deformer network, the dots, the black dot here, and to show the corresponding connectivity, how it looks like in macaque brain. We can then measure the local maximum cross-species similarity based on the gradients. We just name it as Functional Connectivity Homology Index. So this index represents the likelihoods that brain function gradients in one species can be represented in another species. As you can see here, this index showing that very high cross-species similarity sitting in the primary areas. For example, the V1, MT, the primary auditory regions, the FFA regions, even though the FFA regions is not included in our landmark sets. And one thing we noticed that the TPJ, temporal parietal junction, has 0 index, which indicates that we cannot even find the similarity in macaque, similarity functional gradient in macaque as compared to human. This is consistent with literature, as many studies suggest that human TPJ might not exist in macaque. At network level, we find the homology index follows hierarchy, that primary networks showing high similarity, while the high order social cortex, like frontoparietal and deformer network, showing low similarity. We further look into more local regions and show that functional homology also follows the hierarchy in the local regions as well. So for example, the early visual pathway, the circular radiation patterns in the empty regions, the ventral visual pathway, and the dorsal to ventral topology at this symmetry sensory regions. For the subsystem in frontoparietal and attention networks, the functional homology also shows a hierarchy as well. For example, the frontoparietal subnetwork A, which is more relevant in mentalizing and emotion in the literature, showing the low homology index here. And the attention subnetwork A which is highly connected to hippocampus compared to network B in the literature, also shows low homology index here. In deformal network, we looked into 3 subsystems. The core deformal network, which are more associated with self-reference in the literature, showing a lowest similarity of homology score here, and followed by this dorsal medial subsystem, which is more relevant to mentalizing theory of minds. The highest homology score found in deformal network is this medial temporal system, which is more relevant to our memory system. We also correlate the homology score with the importance rank of deformal network subregions based on a recent study. So we find a significant negative correlation between importance of default subregions and this homology index. So this indicates that the most important subregions in deformal network has the lowest cross-species similarity. In other words, the most important subregions in deformal network in human might have developed the latest in evolution and has the recent evolutionary adaptation. So here we show the cross-species similarity map seated in macaque brain and how it looks like as compared to human deformal network. Surprisingly, the similarity pattern of a few deformal network regions in macaque doesn't really look like the typical deformal network in human, but more like a frontal-parietal and attention network. This suggests that macaque may have deformal network, but it is still under development and it's not at the same hierarchy level as compared to a human deformal network. In particular for the dorsolateral frontal lobe, the posterior middle cortex like PCC and brachiornis, the TPJ area, which is the parietal gyrus here. We also link the homology index to the cognition components and find a negative relation here. So the high level cognition components is more likely to locate it in the region that has a low level cross-species similarity. Finally, we demonstrate the evolutionary deformation from macaque to human. For example, the macaque V1 is sitting on the lateral occipital lobe, but in human, it's all in the medial occipital lobe, which means in the evolution, maybe the V1 region has been pushed and squeezed from lateral into the medial and has the higher deformation here. And the expansion from the frontal lobe in human has also pushed the parietal backwards in general. And such expansion can, such surface expansion also follows the hierarchy between networks as well. So taken together, we demonstrate the joint embedding approach to provide a solution for cross-species alignments. We developed the functional connectivity homology index to quantify the cross-species similarity. And this index varies between and within networks in hierarchy manner, which reflects the gradient of evolutionary change from unimodal subsystem and the transmodal system. In addition, our finding provides important view on how the deformal network may have emerged in humans. The deformal network as epic of cognitive hierarchy is a relatively recent evolutionary adaptation. I would like to share the credits with Carl Hinsnani from Gia-Glenn's lab. And many thanks to Mike, Gia, Daniel, Johnny, all my colleagues and collaborators. I also would like to express my special thanks to Daniel Maglis. He's my inspiration for many gradient work. And thanks to HCP Data and all the labs that generously share their data on the Prime DE. And thank Boris and all the organizers again for this workshop. And thank you for your attention.